Good morning, students. Heterotrophic nutrition. What are heterotrophs? Animals and fungi. There are three types of heterotrophic nutrition: holozoic, parasitic, and saprotrophic. Holozoic mode of heterotrophic nutrition involves killing of the entire organism and deriving nutrition out of it. So, all animals do it. Most of the animals that we see. Parasitic mode of heterotrophic nutrition. In this, the organism is deriving nutrition from the other organism without killing it and by taking even shelter from it. That is called parasitic mode of nutrition. There are many parasites like ticks, mice, leeches, tapeworm. These are all animal parasites. And Cuscuta, it is also known as amarbel. It is a plant, parasitic plant. So these are parasitic animals. These are parasitic plants. And coming to saprotrophic mode of heterotrophic nutrition. In this, the particular heterotrophic organism is deriving food from dead and decaying matter. Most of the fungi, they derive the nutrition like this. So example for such fungi are mushrooms, bread mold, which is also known as actually rhizopus. Okay. Coming to the unicellular organisms and their nutrition. There are many unicellular organisms, prokaryotes, all of them are unicellular. Under eukaryotes, unicellular eukaryotes there, like amoeba. In amoeba, like unicellular organism, since the organism itself is single cell itself is the entire organism, the complete surface of that organism is in direct contact with the environment. So, most of the life process, they carried out by simple diffusion because entire surface is in direct contact with the environment. Amoeba forms pseudopodia, which are also known as false feet. These are finger-like extensions that are formed by the protoplasmic movement inside the cell. These pseudopodia, they join by enclosing the food particle, with which food vacuole is formed inside the amoeba cell. Into this food vacuole, digestive juices are released and food will be broken down into simpler substances. Then these simpler substances will be diffused into the cell from the food vacuole. With the undigested material, the food vacuole touches again the cell membrane and it will be just moving out. That is how in unicellular organism like amoeba, the digestion or nutrition is taking place. Paramecium is another unicellular protozoan in which pseudopodia are not formed. Paramecium is covered by many small hair-like structures called cilia, and paramecium takes the food at a particular spot. Food will be directed to this spot by the ciliary movement. Now coming to our large section, plants and animals. Plants take in carbon dioxide and water and glucose is made. Animals take complex food. From that, glucose, amino acids, fatty acids, like these substances are derived. How are they just made? In plants, how glucose is made? In animals, how glucose, amino acids, fatty acids are derived from the complex food. This processing is done by so many biochemical reactions which are catalyzed by specific substances called biocatalysts or enzymes. So these biocatalysts or enzymes, they actually play the role in combining carbon dioxide and water to form glucose or in breaking down the complex food to form glucose, amino acids and fatty acids. In plants, the carbon and energy requirements, they are met by photosynthesis. Carbon dioxide and water are taken in and using the light energy which is captured by chlorophyll, carbohydrates are formed in photosynthesis. These carbohydrates, to some extent, they are utilized and excess will get stored in the form of starch. The storage form of carbohydrates in the plants is starch. The same way, even in animals also, the excess carbohydrates or unutilized carbohydrates are stored in the form of glycogen. That's why glycogen is called animal starch. So if you take the photosynthesis, it occurs in 3 events or 3 steps. The equation for photosynthesis is 6CO2 plus 12H2O gives C6H12O6 plus 6O2 and 6H2O. Right. The 3 events are absorption of light energy by chlorophyll, then conversion of this light energy into chemical energy, and using this chemical energy reducing the carbon dioxide into carbohydrates is the 3rd event. First event is absorption of light energy by chlorophyll. Second event is conversion of this light energy into chemical energy like ATP, because light energy cannot be directly utilized for the reduction purpose of carbon dioxide. So first light energy has to be captured. This has to be converted into chemical energy like ATP, and then using this ATP, the carbon dioxide or CO2 is reduced to carbohydrates. These are the 3 events that take place in photosynthesis. So this is normally occurring in normal plants. In desert plants, wherein stomata are present in pits in order to prevent the water loss by transpiration. How CO2 is obtained into the plants? By stomata. Normal plants, stomata are present on normal surface. In desert plants, stomata are present in deep pits. So generally, during the daytime, these stomata are closed in desert plants. Why? Because excessive temperature will be there in the deserts, which makes transpiration rate to go very high. In order to avoid that, desert plants take up carbon dioxide in the night so that they can avoid much transpirational loss of water. The carbon dioxide that is taken in will be stored by preparing an intermediate. The carbon dioxide which is taken up in the night is stored in the form of an intermediate. And on this intermediate, the light energy which got converted during the daytime into chemical energy will be acting upon, and photosynthesis takes place. Where this chlorophyll is present in the plants? In chloroplasts, which are present in large number in green colored leaves and in the tender parts of the stem where still green color, or that is chloroplasts, are present. There is an activity to prove that chlorophyll is essential for photosynthesis. Already I have discussed it in the previous videos. Okay, wherein what we are going to do, we take a potted plant with variegated leaves. Variegated leaves means different patches of colors are present in the potted plant. We keep this plant in dark room for 3 days. Then we take it out. Why we are keeping it in dark room for 3 days? So that entire starch gets utilized up. Starch is the storage form. So this starch will be getting utilized in this 3 days. Then what are we going to do? We keep this plant under sunlight for about 6 hours, not more than that. So that limited amount of photosynthesis takes place. So that the chlorophyll containing areas only are going to contain starch. If the plant is put for longer time, the starch that is prepared will be just spreading across the entire leaf. So our intention is to prove that chlorophyll is essential for photosynthesis. So we are keeping this potted plant for only 6 hours under sunlight. Then we are cutting a leaf. Then we are marking the green colored areas of this leaf on a paper using a tracing mechanism. And what are we going to do? We boil this leaf. In boiling water, we are boiling this leaf to arrest the metabolic activities. Then we are keeping this leaf in alcohol containing tube, and keeping this alcohol containing tube in water bath, again we are boiling it so that the green color will will be getting dissolved. The chlorophyll will be getting dissolved in boiling alcohol. Why this chlorophyll is not getting dissolved in water? Because it is organic solute, so that it will be getting dissolved in that one only. Right. I already have discussed it. Okay. Why the chlorophyll has to get dissolved? Why you have to dissolve this chlorophyll? If you are not dissolving this chlorophyll, in the next step where we are going to test the presence of starch in the previous green colored areas will not be given, because chlorophyll will interfere in the test. So we are separating this chlorophyll from the leaf. So only we are dissolving it in alcohol, boiling alcohol, by keeping that alcohol containing tube in the water bath. Why you have to keep this alcohol containing tube in water bath? Why not we can heat it directly? Alcohol is highly inflammable. If any direct heat is given to it, even to test tube, it may cause what? The flame. It may cause some fire accident. So if you keep in boiling water bath is not direct flame. So only temperature is given. Temperature alone cannot cause any what? Fire. Fire itself is going to cause fire. Right? Okay. So we are providing heat in water bath and making this chlorophyll get dissolved in that boiling alcohol. And take out this leaf and go with the iodine test. Only some areas you will get a blue-black color formation. Other areas you are not going to. What are these areas? Where are you getting this blue-black color formation? Earlier, where you have marked the green patches, in that area only you will get this blue-black coloration. This shows that chlorophyll containing regions alone could carry out what? Photosynthesis. So this activity proves chlorophyll is essential for photosynthesis. Without chlorophyll, the other areas could not carry out photosynthesis. So there is no starch in non-green color areas of that variegated leaf. Right. Coming to stomata. Stomata are the tiny pores in the surface of the leaves. And stomata are guarded by two kidney-shaped cells called guard cells. And if these guard cells are swollen, the stomata will open. If these guard cells are shrunken, the stomata will get closed. So stomatal opening and closing is the function of guard cells. When water flows inside the guard cells, the guard cells will become perfect kidney shaped and the stomata will get opened. When the water comes out of the guard cells, then the guard cells lose the kidney shape and the stomata get closed. Right. Why opening and closing? Why can't these stomata kept always open? The stomata are always kept open. Continuous transpiration takes place. Transpiration is the loss of water through the open stomata in the plants. Continuous transpiration causes severe water loss to the plant. So, stomata are opened only when there is a need of taking in the carbon dioxide, because through stomata itself gaseous exchange occurs. Taking in carbon dioxide, releasing out oxygen during the process of photosynthesis occurs by stomata only. So when there is a need of taking in of carbon dioxide, then only the stomata will open. When there is no need, like in the night time, there is no sunlight, no photosynthesis can be carried out in the normal plants. So what will happen? They do not open, so that there will not be any intake of, there will not be any water loss during the night time. During the daytime, that too in the normal sunlight, not under excessive heat, what will happen? The stomata will be opened and CO2 will be taken in and photosynthesis is carried out. So like this, the stomata will help in gaseous exchange and transpiration process. And plant closes stomata in the night because there is no need of taking CO2. And when there is no need of taking CO2, stomata gets closed. And stomatal opening and closing is function of which cells? Guard cells. How guard cells will make this process? Whenever stomata are needed to open, water flows into the guard cells and guard cells will become into perfect kidney shaped or bean shaped then stomatal pore will get opened. When there is no need of opening of stomata, then water moves out of the guard cells and guard cells lose the kidney shape or bean shape and they will become shrunken and the stomata gets closed, such that stomatal opening and closing is a function of guard cells. Thank you.